A city is built and maintained by the efforts of many different kinds of workers. The human body is not very different. We need many workers to sustain life. Who are these workers of our body? Proteins. You might think of meat or dairy products when you hear the word protein. Yes, these foods contain a lot of proteins which are broken down and reformed inside our body into various kinds of microscopic workers. For example, the digestive enzymes which break down the food we eat are proteins. Our cells' structures are maintained by cellular skeletons. These are made of structural proteins. Transporter proteins, just like truck drivers along the highway, carry cargo along the cellular skeletons. Hemoglobin, the oxygen transporter protein, is abundant in red blood cells. Antibodies detect uninvited intruders and allow our immune system to fight against them. Antibodies are also proteins. Our muscle movement is possible due to special proteins that move muscle fibers. Hair and nails are also composed primarily of proteins. As you can see, proteins are everywhere in our body, doing almost everything that we need to be alive. Their existence and behaviors are essential to life. There are over 20,000 different kinds of proteins in our bodies. Just one specific protein missing or malfunctioning could lead to disease, sometimes fatal. We cannot live a healthy life without normal sets of proteins. That's why many bioresearchers all around the world try to study the nature of proteins. Until not long ago, the procedures for analyzing proteins were somewhat complicated. First, one must grow many samples, a process that could take from days to weeks. Once the samples are ready, they are killed. The cells are destroyed and the proteins within are extracted. After isolating the proteins from the cell mixture, they are separated according to their size or electrical charge by a process called electrophoresis. Finally, the protein we're looking for can be detected by using its specific detector antibody. This process takes many steps and a lot of time to carry out these experiments. Worst of all, we're only getting a snapshot of how much there was, with limited information about its location within the cell or how it was changing in the living cell. However, in the 1970s, a whole new approach to protein analysis began to develop. It all began with a floating marine animal, the jellyfish known as Aquaria aquaria. The green glow emanating from this jellyfish is caused by a special protein now called the green fluorescent protein, or GFP. Osamu Shimomura, a Japanese-born marine biologist, wanted to find out how the jellyfish produced green luminescence.
일단은 오사모 시모무라 박사님은 이 그린 플로레센트 프로틴을 순수 분리를 하신 분입니다. 맨 처음으로. 그래서 그 단백질이 어디에 있을지는 그 당시로서는 좋은 아이디어가 없었지만은 그 단백질의 존재를 알게 됐고, 그리고 그 단백질, 싱글 단백질, 단 하나의 단백질이 형광을 든다는 것, 아주 좋은 발견을 하셨죠.
But isolating a protein is not enough. Just as a car is produced according to its blueprint, every protein is produced according to a precise blueprint. This blueprint is the DNA gene. In every single cell, there is a cell nucleus, and inside we find DNA, in which our genetic information is encoded. Every protein, including GFP, has its own blueprint encoded in a DNA gene. Douglas Prasher, an American molecular biologist, was the first person to clone and sequence the DNA gene for GFP. Namely, he was the first person to get the DNA blueprint for this protein.
단백질이 있으면 이제 유전자가 있어야 되죠. 그러니까 유전자를 가진다는 게 아주 중요한 바이올로지였어요. 유전자를 가지고 있어야만 우리가 원하는 단백질을 만들어낼 수가 있고, 또 원하는 단백질에 또 컨주게이션도 할 수가 있고, 그러니까 붙이는 거죠. 그렇게 할 수도 있고, 더 다양한 단백질을 만들어낼 수 있는데, 그 유전자의 존재가 아주 중요합니다. 단백질만 있어야 되는 게 아니에요. 그래서 유전자를 클로닝 하신 분이 그 다음에 도글라스 브레이저 박사님이시죠. 그러니까 1992년에 유전자를 클로닝해서 논문을 쓰셨는데, 제가 개인적으로 생각에는 아주 가장 중요한 일을 하신 분입니다. 유전자 클로닝 한다는 게 그 당시에는 쉽지가 않은 일이었고, 그리고 GFP 유전자 클로닝을 함으로써 그 다음에 아주 많은 분들이 아주 좋은 연구를 할 수 있습니다.
So what's the big deal with this jellyfish protein? As we know, proteins are crucial targets of biological and medical research. But we could only study the proteins in an indirect, cumbersome way. With GFP, we became able to study proteins much more directly and conveniently. GFP showed its potential as an excellent marker, or tracer molecule. Let's suppose we want to study a specific protein named alpha. We have no idea where alpha is located inside a cell, where it moves around, or how much alpha is produced. There is no way to see and distinguish alpha directly. Now, here comes the GFP to help out. Alpha is a protein, and GFP is also a protein. Suppose we link these two proteins. Alpha is hand in hand with GFP. Now, if we want to find alpha, all we have to do is find GFP instead. Finding GFP is very easy. We just shine an ultraviolet or laser light and the GFP will glow green. Now we can see inside the microscope and inspect the traces of green light.
네, 그렇죠. 이제 이 그린 플루어센트 프로틴이 나오고 난 뒤부터 우리가 어떤 유전자가 있으면 그게 언제 익스프레션이 되는지, 우리가 어떤 프로틴이 있으면 그게 세포 내에서 어디로 가는지 그걸 정확하게 볼 수 있게 되기 때문에 완전히 그냥 레볼루션 되었죠. 이 바이올로지 분야에서는. 그래서 신경생물학이라든지 세포생물학 여러 분야의 바이올로지 분야에 아주 두루두루 지금 쓰이고 있습니다.